Hi everyone, this is the start of Chapter 20 on molecular technologies. I know not all of you are in the lab, but it's helpful to know these technologies, especially as you're reading through literature. And for those of you in lab, some of these may be things that you've already done and that you have a good grasp on, but it's a nice way to remind yourself of the techniques. So in this first part, we'll talk about gene cloning and using vectors. We didn't actually cover this in lab, but this is something that you are now reading about, at least when you're looking at the coronavirus and building up a cDNA library. So in this one, we'll talk about how we clone genes and put it into an actual vector, how a cDNA is made, and how a genomic library is different than a cDNA library. So gene cloning just refers to when we isolate and then make many copies of one particular sequence of DNA. And this basic method was developed early on in the 1970s, and now we have much more refined methods and a greater ability to do things with the clones that we make of these genes. This table is a great one of kind of summarizing the different things that we'll be talking about, at least in this chapter. It goes into detail on DNA sequencing. Those of you in lab, you have a little bit of experience with this, but certainly from the coronavirus assignment, you now know a little bit more about that. So it's important to have cloned genes because this gives us enough DNA in order to be able to sequence the DNA. If we just have a small sample, we may not have enough to run the actual experiments to sequence it. We can also see that gene editing would be something that could be very helpful. Being able to change a particular DNA sequence means that we could actually look at the promoters and regulatory elements as well as studying mutant genes and mutations in the coding sequence, and that can help us understand a protein structure and function. We also do DNA probes with these gene clones that we do, and this allows us to label DNA strands with a cloned gene if we have probes that were identified from the RNA. This is known as northern blotting, and we'll get into that later in this chapter. And then lastly, expression of cloned genes is one of the ways that we most regularly use them. And so they can be introduced into different cell types or different species. Typically, they're often put into bacteria, into a plasmid, and then this allows expression in great quantities since bacteria grow very quickly. And this can be used, if we have expression of it, in research. This allows us to perhaps see what kind of cellular function a particular gene has, or that coding sequence can be placed next to a very active promoter, which allows us to express lots of the protein, and then we can use that to study the protein. In biotechnology, we use cloned genes to introduce ones into bacteria that can make pharmaceutical products. One of those that's great is insulin, so we can actually get bacteria that will produce insulin by having these cloned genes put into them. In addition, we can do clone genes into plants and humans to make plants and animals to make transgenic species with desirable traits. This is fairly common in plants, things that maybe seedless watermelon or different things like that are used. And lastly, we look at expression of these clone genes in clinical trials of being able to look at gene therapy if we can clone genes that have the right sequence, that maybe those could override ones where we have a mutation. So DNA is involved in these cloning experiments, and the cloning experiments actually is the joining or recombining of two different DNA molecules. First, we would look at our chromosomal DNA, so that is really our DNA segment of interest. If we're thinking of the coronavirus, obviously that would be the segment of the viral genome. Now, in the case of coronavirus, keep in mind it's RNA, so we have to then reverse transcribe it into DNA in order to do this. But we might think about the chromosomal DNA being that one of interest that we're looking at, and that we would go ahead and purify it so that we could use it in a variety of biochemical techniques. The second piece of or kind of DNA we need is a vector DNA, and this is actually what we are going to recombine our gene of interest with. We will use that gene of interest and put it into a vector that will then be able to be replicated into a host chromosome into a host without integrating in the chromosome. So, for example, we may use a plasmid in bacterial cells. It doesn't become part of the chromosome of a bacterium, but it certainly would be replicated within that bacterial cell. Or maybe we'll put it into a virus and then have it replicated in that way. It is what serves as the carrier for the DNA segment that we're interested in. So what do we use as a vector? So the vectors most typically that I've worked with and that you would probably work with in a lab are plasmids. However, we sometimes use virus as vectors as well. These plasmids or virus genomes, what we're looking for is that, and I'll probably stick to the plasmid because it's a little bit easier, but in a plasmid we would want our plasmid to have an origin of replication, and that way we have an area that the host cell enzyme could start replication of this particular plasmid. We also really want to have some kind of selectable marker, and this allows us to know that the host cells that we're looking at carry the vector rather than ones that we maybe didn't pick up the vector. And the way that we usually do this is that our plasmid would contain some sort of antibiotic resistance gene as well, so that we can grow these on medium that has antibiotics in it, and the ones that grow mean that they have that plasmid taken up by them. In addition, we have to have a site on the vector DNA to put in that DNA segment. So there has to be a site that we can allow that DNA segment to go into. And often we think about using a site where that DNA segment will go into, that we can actually recognize that that site, perhaps it's a gene that we recognize the function has been lost, that tells us the DNA went into the right place. And so those are the things that we want as ways that we would look at cloning this sequence. So restriction enzymes are how we actually go about inserting DNA into a vector, and it requires at least 2 steps. In general, the first step is that we digest both our vector, so in the case of a plasmid, we would digest our plasmid, and we would digest our chromosomal DNA. And what ends up happening is that we use the same endonuclease to do this so that we have compatible DNA ends. Once we've cut both of these 2 pieces of DNA, we would then go through joining them through ligation, often using the same ligase that's used in DNA replication. So the enzymes that we're using to digest the DNA, they're either referred to as restriction endonucleases or restriction enzymes. In lab, you guys did this with our salmon genes. You guys were using, we did the HIN3 was the one that we chose as our restriction endonuclease. These will bind to each of the specific DNA sequences, and then they will cut them, the DNA, at the defined locations. So these 2 defined locations, one on each strand. And this is just giving you an image of what this might look like, but sequences are actually recognized by restriction enzymes. So I often think of restriction enzymes as like a pair of scissors, but a specific pair of scissors for just a particular site, some site that they can recognize. And so often these sites that restriction enzymes recognize are called palindromic. That means that the sequence is identical when read in the opposite direction on the complementary strand. And this is giving you an example for the restriction enzyme EcoR1. And what you see is that in the 5' to 3', it's GAATTC, and then we see the exact same on the complementary strand running from that 5' to 3' end. And generally that's what we see with the restriction enzymes, is they have particular palindromic sites that they can cut. And with this cut, think about that phosphodiester backbone of DNA, right? So the rails of the ladder, they can be cleaved in 2 different ways. One of them is what we would call a staggered cut. And what this does is it creates sticky ends, meaning that we have short overhangs between the 2 complementary strands. And those ends then can only be joined with complementary sequences with other sticky ends. The other way that we could do this is blunt ends, meaning that it cuts that double strand at exactly the same point. And I'll show you a quick picture of the staggered, but you can imagine then what the blunt cut would look like. So over here, this is showing that we have 2 different sources of DNA. On the left, we have our blue strand, so maybe this was a strand that we're looking for in our vector, and then over here we have the purple strand, and perhaps this is our DNA of interest. What we're going to do is we're going to incubate both of those with our restriction enzyme. That restriction enzyme is looking for the cut area that it can cut, and in this case, when it cuts that area, you can see there's an overhang, right? So we have these overhangs. It cuts our DNA of interest also with these sticky ends. What's nice about this is these sticky ends are complementary to the other ones. And so what we get is we get an overlap of a few hydrogen bonds, and we can then put these together, well, these will stick together, these sticky ends. Now, before we go ahead and look at this, what we want to do is then use ligase to go ahead and covalently link the DNA backbones. So you can see up here we have the blue from the original maybe vector DNA, and we have the purple from the DNA of interest, as well as on the bottom. Now, if this was to be a blunt cut, what we would see is that this would have cut straight down here at the GC base pairing, and same over here, and then it can actually be added to any other blunt cut just by adding in ligase. So hopefully that makes sense between those two. For the steps of gene cloning, we can see that we're going to purify that chromosomal DNA, right? We have to purify that and then digest it with the restriction enzyme. And then we're also going to purify the plasmid or vector. And once we've purified that, we digest that as well with the same restriction endonuclease. And that will create those ends that are compatible between the two DNA samples. So between our chromosomal DNA we're interested in and the actual plasmid that we're going to be putting it into. I'm going to pause here, and then I'll finish up the second half of this particular mini lecture, kind of going through the making of a genomic library and a cDNA library. If you have any questions, please feel free to ask me.